Thanks very much. I'm really grateful to the organizers for giving me a chance to tell you about work in the lab towards studying robustness in zebrafish development, which is an incredibly robust process. So, early in development, as the cells are sweeping out the lineage and making fate decisions, they're proliferating and growing exponentially. So you can just do a thought experiment where you can ask, okay, suppose that a particular progenitor cell of some clade there's a little bit more of those in a given embryo. What happens to its descendants? Do you end up with a massive excess of those descendants in the final animal? Because if you did, you'd expect to see, major anatomic defects relatively often. And yet rarely do we see, a zebrafish, a fish swimming around with one little fin and one giant fin. That really only happens in Finding Nemo. In fact, embryonic development is incredibly robust. You can remove 30% of the cells in embryos that are 4 hours post-fertilization, and at 26 hours, those embryos will look basically normal, but they'll be a bit smaller. So we are interested generally in how genomes encode these incredibly robust programs of gene regulation and development. It's widely believed that regulatory genes are organized into circuits that canalize cells into fate decisions and into fates in the proper stoichiometry and proportions. These circuits and signaling pathways regulate organ size, and they allow cells to communicate with one another to self-organize into precise patterns during tissue morphogenesis. So generally, we are interested in knowing which tissues and cell types develop most reproducibly across individual embryos, which genes ensure that reproducible development, and how those genes are organized into circuits and signaling pathways that stabilize the output of the overall developmental program. And we want to be able to address these questions looking at every cell type in the animal and every gene in the genome. And so our tool for this is single-cell RNA-seq, which is great because you can make atlases of development like these three that I'm showing you here from the zebrafish. You can find all the cell types. You can count their abundance in embryos. And you can see whether or not they're lost or gained in response to perturbations. And we are interested in knowing how each cell type varies in abundance across embryos and whether or not perturbations alter that variability. But in order to answer that question, we need to be able to look at many, many, many embryos, many replicates, if you will, of the developmental program. And doing that with single-cell RNA-seq is technically and economically challenging. So we've been working on a way to do this, and it's built around a technology we developed last year with Jay Shanduri's lab called SkyPlex, in which, and the way we're using it in this project is we're taking an individual embryo, many individual embryos, and putting each one into a different well, and then dissociating the cells of each embryo into little cell suspensions, and then labeling them with DNA oligos that carry in them an embryo identifying barcode sequence. And then when we pull the nuclei out of those cells and pool them together and sequence them all as a pool, we capture not only the transcriptome of each nucleus, but also the oligo that it was labeled with, so we can tell which embryo that nucleus came from. And in order to do this, we need to be able to sequence, lots of cells. So we're using skyRNA-seq, which is a combinatorial indexing-based method we worked on with Jay a few years ago that can allow you to profile millions of cells in a single experiment. So having done that, we can just count how many cells there are of each type in each embryo and compare these counts across time or in response to perturbations. So to get a sense of the baseline variability in each cell type in the embryo, we first created a time-resolved reference atlas of zebrafish development. And so we've sequenced a little bit less than 1 million cells from 859 different embryos, and these were collected at one of 15 time points from 18 to 96 hours post-fertilization. So this window opens when segmentation is ongoing and semitogenesis is happening, and goes all the way through embryogenesis and into the early larval stage. We can tell based on looking at marker genes which tissues are represented, and we can see that the cells come from 33 different tissues and are one of 85 major cell types. So up until this point, this looks like a sort of a usual cell atlas, but the distinguishing feature is that we know which embryo each cell came from. And we can tell that we're sampling about 1,000 cells per embryo. It's not completely even over the time course because we're devoting more sequencing to certain time points than others, but that gives you a sense for how much we're sampling from each individual. Now, how are the genes in the genome, the regulatory genes, organized and regulating one another in order to stabilize the output of the developmental program? So to address this question, we're looking not only at the wild-type embryos that I just showed you, but also many mutant embryos. And we're looking at embryos that are, have any one of these genotypes where these lineage, regulatory factors that I'm showing you are knocked out. We're targeting genes that are important in the mesoderm and the central nervous system, and then a few in the neural crest as well. Most of these are transcription factors, but we've got a couple of morphogens and the receptors in there too. And we're doing this with CRISPR. We're doing this by taking advantage of some recent advances in efficient CRISPR mutagenesis, where we're targeting each of those genes with 3 independent guide RNAs and then co-injecting the guides along with RNPs into one-cell stage embryos. And what this allows us to do is make F0s, sort of at scale. We can do a lot of these in a day, and then sequence the F0s with Skyplex. And another advantage is that, in contrast to conventional mutants, for at least some of these backgrounds, is that a very high proportion of the F0s have the expected phenotype. So this is a scalable approach. The one downside, which is important to keep in mind, is that we have to accept some amount of mosaicism in these embryos, and that'll be important later. But we have, as controls for that, we've got for a bunch of these genotypes, conventional nulls as well. Okay, so now I'm showing you cells from the mutants, and this is 3 million additional cells collected from each of 812, or one of 812 embryos. Those embryos are from one of the 22 different genetic backgrounds I showed you a moment ago. And if this data looks exactly the same as the previous data set, that's just because we've projected these mutant data onto the reference time series using an algorithm very similar to the azimuth algorithm. Okay, so I'm not going to be able to show you more than just a little peek at the data, but I want to tell you about this vignette from the neural crest. So the neural crest cells are a transient and migratory cell population that some refer to as the 4th germ layer, because these cells migrate out in development all over the embryo and give rise to a bunch of different cell types, as you can see here. And we're targeting 2 transcription factors in this experiment, TFAP2A and FOXD3, both of which are transcription factors important in neural crest cells. And loss of function mutants, single mutants for either of these genes, lead to depletions in pigment cells and other cell types that emanate from the neural crest. And in the double mutant, there's basically no neural crest output. The cells have no pigment, the embryos have no pigment cells, etc. So these are the single cell data for the double mutant. I'm coloring each cell type by whether or not there are more or less cells in the double than there are in control embryos at matched time points. And what you can see is that, as you'd expect from looking under the microscope, there's sharp depletions in the clusters corresponding to melanocytes and xanthophores and the pigment progenitor cells. And there's also a reduction in craniofacial cartilage, which is consistent with this jaw phenotype that the double has. You also see changes in cell types that are harder to spot under the microscope. So here's another view of the same data. I'm just going to show you box plots now of the number of cells of each type across the embryos. And in contrast to controls, you can see that there's really not a reduction in pigment progenitors in TFAP2A single mutants, whereas in embryos lacking FOXD3, they're sharply reduced. And if you look at the differentiated pigment cells like melanocytes, they're altered in the single mutants and completely ablated in the doubles. And this is true for the other 2 pigment cell types as well. We see changes in glia and actually increases in glioblasts, which is interesting. And so we're excited to follow up on that. It's a little bit challenging to make inferences about this population, though, because glia come from the neural crest, but also from other places. We do see the expected reduction in sensory neurons. And so overall, the phenotype that we arrive at by counting the number of cells per embryo for each of these types is totally consistent with what we'd expect although there are some new observations too, such as we see a major increase in spinal cord neurons. And that's cool because spinal cord neurons are not generated by the neural crest. So we'll have to figure out what's going on there. And to do that, we're going to have to look much earlier than 18 hours post-fertilization. Okay, so you may have noticed in addition to changes in the mean, there appear to be some changes in the variance in the number of cells per embryo of some of those types. So how do we assess whether that's real? Well, if we want to test whether or not a perturbation significantly alters the variance in the number of cells of a type, we have to confront and address this issue that's very common in count data, which is that the mean of a thing that you're counting, like a cell type, is related to the variance of the thing that you're counting. And so, for example, in this cartoon, each dot is a cell type, and the horizontal axis encodes the average number of cells of that type per embryo. The y-axis is the coefficient of variation, the standard deviation over the mean. So you can see this trend here with your eye, and we can capture it with methods from, say, bulk RNA-seq analysis. We can fit a generalized linear model relating the mean to the coefficient of variation, which we can model as a gamma distribution. And then we can also describe the error in where each cell type is in this plot by fitting a beta binomial distribution to each cell type's counts across embryos. And that gives us a little confidence interval for each cell type. So now we need only ask, if we want to know, is a particular cell type more variable than one would expect for, the typical cell type of that abundance. We just need to ask whether or not it's above the curve enough to escape its own confidence interval. Likewise, we can ask whether or not perturbations elevate the variance enough to be unexpected. And perturbations often change the mean too, and we can account for how much the variance typically changes when the mean changes. And sometimes what you'll see is that a cell type is reduced in abundance and its variability is kind of brought back in line with expectation. Okay, so with this sort of model in place, we can now color our box plots by how variable the cell types are across embryos in each genetic background. And in doing that, we can see that, although the average number of pigment progenitors in TFAP2A mutants is basically the same as controls, those cells are more variable. We can also see this trend much more prominently when we look at melanocytes. And overall, I think we can say that in addition to loss of TFAP2A or FOXD3, changing the average output of the neural crest, losing those genes makes it more variable. All right, you can look at covariances too. You can here I'm showing you that melanocytes are positively correlated with xanthophores in wild type embryos, which is not surprising because they are siblings in the lineage. But if you look at TFAP2A mutants, that correlation is inverted. And that suggests to me, and this is speculation, but it's cool, that suggests to me that we've somehow changed the canalization of this particular fate decision across embryos. One of the other awesome things about zebrafish as a model for development is that they develop externally, so you can perturb them through the environment. And if you raise zebrafish at the reference temperature of 28.5 degrees, you usually get fish that are like straight as an arrow, like this. But if you dial up the temperature even a little bit and still within the physiological range, the range that they get in their natural habitat, you start to see with some frequency embryos with bends in their tails. And if you look closely, they have other anatomic defects as well. So we wanted to understand the cellular and molecular basis of this anatomic variability. And towards that, we've sequenced the transcriptomes of another 700,000 or so cells from 288 different embryos. And these are all wild type embryos, but some of them were raised in hotter water. Now, we want to study the basis of the variability, but in order to do that, I need to tell you about another thing that happens when you turn up the temperature, which is that development goes faster. So if you raise embryos, if you look at a 24-hour-old embryo that is in 28-degree water, it'll look like this drawing on the left. But if you were to do that at 34 degrees instead, the embryo will only take 18 hours. Will only need 18 hours to look like that. So that's a 6-hour acceleration. That's a lot. And we know we need to sort of account for this somehow when we're analyzing variability in response to temperature stress. And the way we've done that is we've created a matrix where the rows are cell types and the columns have the abundances of those cell types in each of the individual embryos. And then we just feed this matrix into a trajectory analysis. So you just put this thing into UMAP and you get the picture like I'm showing on the right, where the dots are embryos and they're following this linear trajectory through the space of cell frequencies according to time. But what's cool is you can sort of facet the data and look at the embryos from any one time point, and you'll see that the embryos that came from hot water are always a little ahead of the curve. And that's true of whatever time point you look at. So we can capture this acceleration in the cell frequency data. And we know from, like Chuck Kimmel's classic paper on how to stage fish using the microscope, we know how much acceleration to expect at any given temperature. We know we have a quantitative model for that. So we can compare that expectation with the acceleration we see in the cell frequency data, and the two methods agree very nicely, which means that we can stage our fish just with the cell frequencies alone and match them up when we're looking at how they vary in response to temperature stress. Now, it's true that dialing up the temperature accelerates development, but that's not to say that all the cell types are affected equally. Some cell types are more accelerated than others in their development. And we learned this by looking at the transcriptomes of cells from hot water embryos and comparing them to transcriptionally similar cells from embryos in reference temperature water, and then just looking at the difference in actual age. So when we do that for each cell type, we can see that some cell types, like the notochord, which is an incredibly important patterning cell type, it really helps set up the anterior posterior axis of the embryo. It's much farther along than other cell types, even in stage matched embryos. So not only does dialing up the temperature accelerate development, it introduces asynchrony in development, and that in turn could drive variability across individuals. So which cell types are more variable at higher temperatures? Well, to find that out, we turn to our mean versus variance plots here and picked out a few that are unexpectedly variable and even more so at higher temperatures. And what we can see, like a good example is the pigment progenitors. And what we can see when we look at these guys is I'm showing you just the number of pigment progenitor cells per embryo spread out over developmental stage on the x-axis. What you can see is that there's not really a big change in the average number of pigment progenitor cells as you dial up the temperature, but there is a big dilation in the variability, and it's particularly pronounced, sort of on the earlier end of development. So you can kind of see that a little bit more clearly in this little heat stripe here. Now, those are pigment progenitors, which give rise to 3 different pigmented cell types. So what happens with that excess variance? Is that like transmitted down the lineage? Are the differentiated cell types more variable as well? Well, it looks like they are. If you look at the 4 cell types, you can see that at elevated temperatures, all 3 pigmented cell types have excess variance at that same stage in development than at reference temperature. And it's to varying extents. Melanophores are more affected than xanthophores, for example. But interestingly, in all 4 cell types, it appears to be kind of tapered off or damped down as the embryos develop further. Okay, so I've only given you a glimpse of this totally huge data set, but I think we can draw a few conclusions. One is that I told you about how we've used Skyplex to sequence around 5 million cells from more than 2,000 individual embryos, many of which have been perturbed in various genetic or environmental ways. Overall, this is a way that you can accurately phenotype or stage individual embryos based on the cell type frequencies. You can do that, and you can also detect shifts in the abundance of each cell type in the embryo, and also sometimes see changes in the variability in those cell types across individuals. We can see that losing lineage determining factors leads to losses and gains of individual cell types, and sometimes increases the variance in the output of a lineage, like neural crest in the case of TFAP2A and FOXD3. Temperature stress accelerates embryonic development of many, but not all, cell types, and that introduces asynchrony, which in turn could contribute to variability across individuals. And it also increases variance in the relative abundances of a number of cell types. So this is what we've learned so far. This is definitely an ongoing project, but I'm very excited about the future. And because this is a computational session, I thought I would highlight what it means for our lab, what this kind of experiment means for our lab, and what new problems we might be able to work on. So, I didn't really talk about this at all, but we have many different genetic backgrounds in this first experiment, and they often phenocopy each other in terms of the cell types that are gained and lost. So I'm hopeful that we'll be able to look at these phenocopying relationships and systematically and automatically maybe assemble the genes that we're targeting into interaction networks, which would be very important for and helpful for understanding how fate decisions are being made and how development is being stabilized. I just showed you like 5 million cells worth of single cell RNA-seq, and I don't think I showed you the expression of a single gene in the whole talk. But many of these methods, I think, could be readily applied to look at variability in transcript levels rather than cell counts. And if we did that, we might learn something about transcriptional regulation within the cells in each cell type in the whole embryo. And I did tell you that there's a strong covariance structure and that you can mess with it in these data with genetic experiments. I think that this could be a rich source of information for understanding the lineal relationships between the various types of cells in the embryo. So these are cool problems in what I think of as like statistical developmental biology, and we're going to be working on them in my lab over the next, I don't know, while. It's going to take us a while. These are hard problems. So I want to conclude by thanking Lauren Saunders, Mike Doherty, and Sanjay Srivatsan, who did these experiments and have been analyzing them together with Maddie Duran. And we've been supported in our team by Brent Ewing, who's a software engineer. None of this would have been possible without the assistance and interpretation and design guidance, and in some cases, hands of 3 other labs, Cecilia Moens, David Kimmelman, and Dave Rabel, who are experts in many of the genes and tissues and cell types that we're targeting here. I want to thank members of their labs and ours. I also want to thank Nola Klemfos from the VBI and Frank Steemers from Illumina for sequencing support in this project, which was funded by the Paul Allen Frontiers Group and the NIH. And I'd like to thank you for your attention, and I'll take any questions if there's time.
Thank you very much, Cole, for this amazing talk. We have a question from Arjun Raj. Very cool. What do you think? Does heat or stress induce variability or reveal cryptic variability?
Well, that is a terrific question. I'll just give you a couple of things that we're looking at to get some more mechanistic insights as to what might be going on. We are very interested in the overall sort of thermostability of the proteome. And we know that different cell types in the embryo have as their kind of repertoire more or less thermostable proteomes. And we're sort of trying to correlate that with how much, with which ones are more or less variable. But we also can see that temperature changes the rate of proliferation. We think it changes the rate of proliferation in certain cell types and maybe not others. So, it's hard to say whether or not this is variability that's being, that would be cryptic otherwise. But I think that there's going to be many possible mechanisms, and a number of them will be true, and they won't necessarily be mutually exclusive, if that makes sense. I know this is a subject, Arjun, close to your heart. We love your review on this area from a couple of years ago, so I figured you might be into this kind of thing.
Cole, the next question from John Murray. Really impressive work. Can you see any cases in your mutants where the cell frequency differences are accompanied by observable differences in trajectories, suggesting novel differentiation paths?
So that's a really good question. And by the way, great to, sorry, I'm not actually looking at you, John, when you're asking this question, but it's great to hear from you. So I was going to show this. We can see a clear case. I just didn't have time. We have a bunch of mutants that target muscle development, and you can see very clearly that some of the mutants for like downstream sort of mid-range effectors cause like sort of slowdowns in muscle development. Cells kind of get blocked halfway through the trajectory, and then if you look at their upstream regulators, or if you look at genes which kind of collaborate with those you look at doubles, for example, the cells are like blocked completely at the entry point to the trajectory. So you can see, barriers very, very easily. And it's a super high, you could see the effect size from space. We have, I'm not sure that we've seen like cells going completely off in a new direction yet. That's something that we want to do. But one of the more generally, and this gets at a comment that Arjun made in his talk, this discretization that we're doing into cell types has some pros and cons. One of them is that, it makes things simple and you can use all kinds of statistical tools, treating things categorically that it's hard to do in the same way with a continuous picture of the data. But on the other hand, it makes it more challenging to do analyses like I think the ones that you're suggesting. So we're taking kind of a first pass through the analysis where everything is in discrete cell types. And then I think in places we'll want to zoom in and take a more nuanced view where something like that might become more evident.
Thanks, Cole. There's time for one more question, so I'll just combine two methodological questions. Dihu asks, why are you using siRNA-seq instead of 10x? And Chris McGinnis asked whether it would be possible to use just a more concise set of 100 lineage identifying genes instead of whole transcriptome sequencing. Because you're interested in tabulating the cell types rather than cell states.
That's a great question. So I'll address the first one first. I mean, the most basic answer is that skyRNA-seq is like much, much cheaper than 10x. Just the library prep is just much, much cheaper, especially because, we're doing it with, it's very DIY, right? It's in-house. So that's one thing. And we need to be doing millions of cells in order to be able to reach the statistical power that we need for these kinds of analyses. So that's just a kind of a power and economics issue. The other thing is that our hashing strategy relies on being able to fix nuclei in order to do these. So this is all single nucleus RNA-seq, and that's sort of easier to do with our way with skyRNA-seq than it is with other tools. So that's one reason. And then the second reason was, why not kind of a targeted approach where we're reading out say 100 genes instead of the whole transcriptome? And I think that there's a strong argument for doing that if you're looking, if you're studying the genes that govern one lineage and you really have highly reliable markers that you can kind of interrogate. That might be great because it would dramatically cut down on the sequencing cost of the experiment, and you might be able to kind of ignore lots of cells from tissues that you don't expect to be affected and whatnot. And I think in the future that'd be a great strategy, but since this is the first time we're doing this, we want to just look at everything.
There's more questions, but I think we could put those in the panel and check there.